Aloha. So today I'm going to share with you research that we've done in our lab. And this research was done actually by a former high school student who was an exchange student during the summer. And he worked in our lab, and we did a simple experiment on rooting of Melanthera integrifolia. So we looked at the rooting response of 3 accessions of Melanthera integrifolia and how it responds to the application of IBA or indolebutyric acid. So in Hawaii, the use of native plants has been promoted in the last 20 years, and Melanthera integrifolia is one of those species that is being promoted as a native ground cover. Although it's been in the market for a while, there has been little to no research being conducted to develop selections of Nehe for particular purposes. Looking at propagation, Nehe is very easy to propagate, and it doesn't require rooting hormones. What we wanted to find out in our study was to look at accessions and see if there is a difference between rooting of accessions and if each accession will respond differently to rooting hormone application. In addition to those differences, we also compared a visual scale of rooting, which is the rooting index, versus WinRHIZO, which is a digital analysis software which actually collects better data in terms of rooting morphology. So the accessions we evaluated in the study were Koko Head from Oahu, Makapuu from Oahu, and South Point from Hawaii. So we collected 80 stem cuttings of these 3 accessions from garden-grown stock, and we graded them based on size. These cuttings were then treated either without or with 3000 ppm indolebutyric acid and then allowed to root for 2 weeks. And then after 2 weeks, we carefully removed and washed all of the cuttings and then collected percent rooting and rooting index first. And then after that, we scanned the cuttings, saved the scanned images, and then analyzed these images in WinRHIZO. So the data that we collected in WinRHIZO were root surface area, root volume, root length, as well as the number of root tips per cutting. All of these data were then analyzed as a factorial analysis of variance in statistics 10, with the first factor as the IBA treatment and then the second factor as the accession. So what we found out is that IBA application improved some of the rooting morphology of these cuttings. We particularly saw an increase in surface area, in root length, as well as the number of root tips of cuttings regardless of accession. So IBA improved these characteristics compared to the control treatment. In addition to that, we also saw some differences between accessions in terms of surface area, wherein we saw the Makapuu accession exhibiting the highest surface area of roots compared to South Point. And you can see here in this poster that there's a sample picture of IBA treated versus untreated cuttings, and you can see some slight differences that actually the WinRHIZO software detected, whereas the rooting index did not. So in conclusion, we found that Melanthera, although it is easy to root, IBA application can improve root morphology. There was some differences in terms of surface area between accessions, with Makapuu having a larger surface area of roots compared to the South Point plants. And we also found that, well, the Makapuu plants were generally bigger and more vigorous compared to the South Point plants. Then finally, we saw that rooting index may be helpful, but as a quick assessment for rooting, but the digital image analysis was a little bit more better in detecting slight differences that the eye cannot see.